So, hi everybody. Let's go ahead and on the next page in our journals, we'll draw this picture into our journals directly. And it will be a picture of an animal cell. And remember, the basic structure is kind of roundish. So, kind of roundish will do. And that was the cell membrane that we drew. So what we're going to do is we're just going to draw this. We're not going to write the functions of all the organelles down again. You can look on the previous page underneath the flaps on the foldable to get the functions of these things. So let's go ahead and put the nucleus in. And remember, the nucleus has got a nucleolus. Function of the nucleolus, this is where ribosomes are made, remember? And then let's call this the nuclear membrane. And let's go ahead and put some chromosomes in, just to remind ourselves that this is where they are. If you draw and then use the same color for the writing as you did the drawing, that will allow you to be able to skip drawing arrows that point to those things. Then we'll put the ER in here, and which is a big network of tubes. Remember, endoplasmic reticulum. In the endoplasmic reticulum, this is produces substances that the cell needs. Some ribosomes could be attached, so let's put some in there. These little circular things. Some ribosomes could be attached. And the ER helps the ribosomes make protein. Okay, then let's go ahead and, for my orange, put a couple of mitochondria in. You can put as many as you want. Mitochondria. Remember, their function is to convert energy in food to energy that the cell can use. Let's go ahead and put in a Golgi body. Golgi body can look an awful lot like the ER. Golgi apparatus, or I think your book calls it the apparatus or the Golgi body, responsible for packaging and distributing materials from the ER. So typically they're kind of close to one another. I'm going to put some lysosomes in here. Lysosomes, their job is to break down large food particles into smaller ones. And they break down also worn out cell parts. So they're kind of like the garbage collector. When parts of the cell wear out, they handle those, take care of it, get rid of them. Let's see here. We forgot, oh no, we put the cell membrane in. So let's go ahead and put a vacuole in. I'll use usually vacuoles in animal cells. are a lot smaller than in plant cells. Remember, the vacuole stores water, food, waste, other things. So that's the storage area. So let's see here. We have the cell membrane, nucleus, no chloroplast in an animal cell, mitochondria. We have the no cell wall, nuclear membrane. Yeah, we have that. Vacuole, ribosomes. We have some ribosomes. Remember, their job is to produce proteins. We have the ER, we have the nucleolus up there. Nucleolus. Ribosomes are also made in the nucleolus. We have the Golgi. Oh, and then everything else. I'll use, let's see here, what color haven't we used yet? Everything else, this, you know, all of these organelles aren't just floating in space, in cell space. They're all set in a jelly-like stuff called cytoplasm. So everything else around here is going to be cytoplasm. Scientists for the longest time believed that cytoplasm was just nothing more than jelly-like material that held these organelles so that they didn't flop around all over the place. But in the past several years, scientists have realized that there's actually a lot of chemical reactions that happen in the cytoplasm. So that's important too. And we've got lysosomes and we put our chromosomes in. Very good. There's our animal cell. Reminder, no chloroplasts and no cell wall. Very good. See you later.